In this PowerPoint, I'll be covering inherited disease. I'll be covering the roles of DNA, mRNA, tRNA and ribosomes in protein synthesis, the effects of point mutations, the inheritance of phenylketonuria, the inheritance of gender and the inheritance of sex-linked conditions, the roles of chromosomal abnormalities in inherited disease, the process of genetic screening, the appliance of DNA recombinant technology in the treatment of diabetes, moral and ethical issues raised by DNA recombinant technology. Protein synthesis. Protein synthesis is the process that your cells undergo to create protein molecules. There are two main stages of protein synthesis. These are called transcription and translation. During transcription, the DNA double helix is unzipped to expose just one strand of DNA. The free nucleotides then produce a strand of mRNA that is complementary to the original strand. Translation is the stage where the mRNA leaves the nucleus and enters the cytoplasm of the cell. It then attaches to a ribosome. Then, the ribosome reads the codon sequences of the mRNA so that the tRNA molecules can bring the correlating amino acids. Point mutations. Point mutation is where a nucleotide base is changed, added, or removed. There are three types of point mutations. These are silent mutation, missense mutation, and nonsense mutation. Each codon codes for a specific amino acid. A silent mutation is where a base is changed but still allows for the codon to code for the same amino acid. Silence mutations are for some species helpful and allow cells to create proteins more effectively. However, for humans, they have been known to cause diseases such as Hirschsprung's disease, Seckle syndrome, and phenylketonuria. Missense mutations are where a letter of the codon is changed and it now codes for a different amino acid. Nonsense mutations occur when a letter is changed and the codon becomes a stop codon. This table can be used to work out which amino acid a codon translates to. An example of a disease that is caused by a missense mutation is sickle cell anemia. It is caused by an adenine molecule replacing a thymine molecule in the template strand of the gene, which will then code for uracil rather than adenine in the corresponding mRNA. The mutated codon will then code for valine instead of glutamic acid. A nonsense mutation occurs when a base is changed and the codon becomes a stop codon, meaning translation stops at that point. Nonsense mutations have been known to cause diseases such as muscular dystrophy. Phenylketonuria. Phenylketonuria is a disorder that comes from a mutation in the phenylalanine hydroxylase gene. Newborn babies usually appear healthy and symptoms arise when the condition is not treated properly. Some symptoms of phenylketonuria include eczema, autism, seizures, and motor deficits. If it is left untreated, then it can cause irreversible mental disability. Introducing a phenylalanine restricted diet soon after birth can prevent a lot of the neuropsychological difficulties. However, maintaining this diet can be very challenging. Phenylketonuria can be detected from a blood sample taken in neonatal screening tests. Phenylketonuria is an autosomal recessive disorder. People can be carriers of phenylketonuria without having it themselves. If both parents are carriers, there is a 25% chance that their child will have phenylketonuria. There's a 50% chance that their child will be a carrier, and a 25% chance that their child will be unaffected. Both parents must have contributed a mutated gene for their offspring to have the inherited condition. The gender of human beings is decided by their chromosomes. Females have XX chromosomes, whilst males have XY chromosomes. The Y chromosome plays a key part in deciding the gender due to the presence of the SRY gene. The SRY gene is located on the short branch of the Y chromosome. The SRY gene is responsible for starting the process that causes a fetus to develop male gonads rather than the female reproductive structures. The inheritance of a sex-linked condition. Sex-linked conditions are often referred to as X-linked conditions. This is because they usually come from a mutation on the X chromosome. With these conditions, males are more likely to inherit them because they only need to receive one recessive mutated allele from their mother to be affected. A female would need to receive a mutated X-linked allele from both parents to be infected. Females can be carriers if they receive one recessive mutated allele and one dominant wild-type allele. An example of a sex-linked condition is haemophilia. Haemophilia is a bleeding disorder where a person's body lacks the ability to clot blood at the normal rate, which can result in excessive bleeding. Most people that suffer from haemophilia are male. This is because the gene that is associated with this condition is located on the X chromosome. Men only have one X chromosome, and one copy of the mutated gene is sufficient to cause a person to have this condition. Men cannot pass this condition down to their sons, as their son will only inherit their Y chromosome from their father. In females, the mutated gene would have to occur in both of their chromosomes for them to inherit the disorder. Chromosomal abnormalities. Aneuploidy is where the number of chromosomes is not the exact multiple of the haploid number. Aneuploidy occurs during meiosis when pairs of homologous chromosomes fail to separate. The scientific term for this is non-disjunction. The loss of a chromosome is called monosomy, whilst gaining a chromosome is called trisomy. Aneuploidy can either result in a baby being born with a disorder such as Turner syndrome, which is X mesonomy, or Down syndrome, which is trisomy of chromosome 21, or it can be lethal in the first trimester and lead to an unviable fetus. Down syndrome is caused by trisomy of chromosome 21. Trisomy 21 is the most common trisomy in a newborn. The additional chromosome rarely comes from the father, and the risk of a couple having a baby with Down syndrome increases as the mother gets older. Women with Down syndrome have a 50% chance of passing it down to their offspring, however many of the affected fetuses are spontaneously miscarried. The majority of men with Down syndrome are infertile unless they have mosaic Down syndrome. Mosaic Down syndrome is where a person has a mixture of two types of cells, and some will contain a normal 46 chromosomes and others will contain 47. Down syndrome can be diagnosed before birth by ultrasonography of the fetus or by blood tests of the mother, chronic villus sampling and/or amniocentesis, or, after birth, by the appearance of the baby accompanied with a blood test. Genetic screening. During genetic screening, a person's skin or blood is analysed to see if their genotype puts them or their offspring at risk of a disease or illness. The purpose of genetic screening could be to confirm diagnosis, advise the likelihood of other family members having the disease, to test if offspring are likely to inherit the disease, and to aid research into the severity of the disease. Pre-symptomatic screening is where people are tested if their health could be in danger. Screening could be for diseases such as cancer. Carrier screening is to see if a person is a carrier of a certain genotype that could put their offspring in danger of inheriting a disease such as phenylketonuria or cystic fibrosis. There are multiple ways that your sample might be collected for genetic screening. Some of these include a blood sample. This is where a doctor or nurse could insert a needle into your vein. For newborns, this is done by heel prick testing. They might also take a cheek swab. Or there's amniocentesis. This is usually for prenatal checks. It's where a doctor inserts a thin, hollow needle into your uterus to collect a sample of amniotic fluid. And there's chronic villus sampling. This is where a doctor takes a tissue sample from the placenta. This could be done through a catheter inserted into the cervix or through the abdominal wall using a thin needle. DNA recombinant technology. DNA recombinant technology is the process of modifying genetic material outside of an organism to either enhance their characteristics or to achieve a more desired characteristic. First, using enzymes, the gene or other piece of genetic material will be inserted into a circular piece of DNA called a plasmid. It then forms a molecule of recombinant DNA that is made from fragments of other pieces of genetic material of interest, joined together with DNA ligase. The plasmid is then inserted into bacteria and antibiotic selection is used to determine which bacteria took up the plasmid. Once we have identified which bacteria took up the plasmid, we can release a chemical signal that instructs the bacteria to continue making the target protein. DNA recombinant technology is particularly beneficial to people suffering from type 1 diabetes. This is because insulin is normally produced in the pancreas so that it can travel through the body, binding to cells and allowing them to take in glucose. A person with type 1 diabetes would have either damaged or destroyed cells in their pancreas, meaning that it's unable to produce insulin as it should, so they need to inject it to prevent their blood sugar levels from becoming dangerously high. In the 1960s and 70s, laboratories were developing human insulin and it was approved for pharmaceutical use in 1982. This means that people suffering from type 1 diabetes no longer need to use the animal insulin that was previously offered. Moral and ethical issues raised by DNA recombinant technology. DNA recombinant technology has vastly improved the treatment options for diseases such as cancer, diabetes, and other genetic diseases, as well as disorders observed in plants. However, further research is required into the safety of genetic engineering and whether it can cause dangerous health implications. Some people are also concerned that genetically engineered plants can crossbreed with wild plants, meaning that the genetically engineered plants are contaminating our biodiversity. Another ethical concern is whether we are creating a new or different life than what was intended, and whether it is actually our place to be doing that, or should we just leave it all down to nature. There could also be safety concerns around using DNA recombinant technology because we could accidentally infect somebody with a dangerous disease. We could try and fix a mutated gene and make it worse than if we had left it alone. There are also privacy issues with DNA recombinant technology because it allows people to find things out about you that they wouldn't typically know, such as if you're at risk of a certain disease. This could pose as a risk when taking out insurance policies, as they may refuse to insure you knowing that you carry certain genes.